Hi class, this is our second meeting, and for today's discussion, we will be discussing or talking about nucleic acid, the DNA and the RNA. For a quick recall, genes are our units of inheritance, and this is composed of the DNA. The DNA can be replicated so that after each replication, after each cell division, the daughter cells will have the exact same copy of the genetic material. Aside from that, there is a flow of information from the DNA to the RNA to polypeptides. For the RNA, it could either be one of the three, messenger RNA or mRNA, a transfer RNA or tRNA, and then to polypeptides. So this is what we are going to talk about for this discussion. For the outline of the entire topics, this lecture is relatively short. We will be covering the structure of the DNA and the RNA after we discuss some of their functions and properties. We will also take a look at the nitrogen-containing bases of these molecules, and then the Watson and Crick model of the DNA, the double helix model proposed by Watson and Crick. And then towards the end of the discussion, we will take a look at the unusual structures that the DNA and the RNA can exhibit, which could be based on their sequences. For the function, main function of these molecules is to store, transmit, and express hereditary information. Just like carbohydrates and proteins, nucleic acids also exist as polymers known as polynucleotides, and its monomeric units are what we call as nucleotides. Other functions, nucleotides participate in energy metabolism. So there's ATP and ADP, which we will be discussing in our next discussions of metabolic pathways. You will see that these are involved as principal donors and acceptors of phosphoryl groups. These nucleic acids also participate in protein synthesis, as you will see later on in our discussion of the central dogma of MolBio. And then they also participate in the regulation of enzyme activity, an example of which is ATP-dependent phosphorylation of key metabolic enzymes. So ATP-dependent meaning when in the presence or absence of phosphoryl groups, these enzymes could be active or inactive. And then they also participate in signal transduction pathways. Let's take a look at the structure of nucleotides. Each nucleotide monomer includes a nitrogen base, so that's number one. So you will see here a purine or a pyrimidine base, and then a sugar, a pentose sugar, and the phosphate group. Just a second. Okay. In this structure, you will notice here prime numbers. You will see here 1', 2', and then 3', 4', and 5'. So what do these prime numbers refer to? These prime numbers refer to the carbons of the sugar moiety, so the pentose sugar. The carbons are numbered this way, 1', 2', up to 5', so that we will be able to distinguish it from the carbons of the nitrogenous bases. Later, you will see that the nitrogenous bases will also have carbons, and these prime numbers let us distinguish which ones are we referring to when we are looking at the carbons of the nucleotides. Aside from that, in this structure, you will also notice the hydroxyl group at the 2' position. So you will notice that it is in pink. This is to take note that in the DNA, in this position is a hydrogen atom and not a hydroxyl group. If you look at the structure, the DNA and the RNA share chemical similarities, but there are also major differences between the two. First, you will notice, like I have said, you have the sugar component, which are different from the DNA and the RNA. For the DNA, the sugar is deoxyribose. You will notice that here, okay, just here, deoxyribose. So at the 2' carbon position, it has one less hydroxyl group. Hence, it is called deoxyribonucleic acid. If you take a look at the RNA, at the 2' carbon position, there is a hydroxyl group. It is a ribose sugar. It contains a hydroxyl group linked to the 2' carbon. That's why it's called ribonucleic acid. And this absence of the 2' hydroxyl group in the DNA, further it increases its resistance to hydrolysis. So that's why it's important. Relative to the RNA molecule, you may have heard that the RNA is more susceptible to hydrolysis and RNA is less stable when you compare it to the DNA. So you can attribute this to this differences between the DNA and the RNA. Second major difference between the DNA and the RNA is the bases that are present in the DNA and the RNA. For the DNA, it is composed of A, G, C, and T. So you have adenine, guanine, cytosine, and thymine. But for the RNA, uracil is in place of thymine. So you have adenine, guanine, cytosine, and uracil. So A, G, C, and U. Third major difference is the strand. DNA is double-stranded while RNA is single-stranded. This difference in the strands of the DNA and the RNA also contributes to their stability. So aside from the hydroxyl group, which is present in the 2' carbon position of the ribonucleic acid, RNAs are also single-stranded molecules, so it is relatively less stable when we compare it to the DNA. Next, let's take a look at the nitrogen-containing bases. So we have the purines and the pyrimidines. So these are heterocyclic compounds with rings containing nitrogen and carbon. So take a look at the structure, heterocyclic rings. So for purines, these are the adenine and the guanine, which contain a pair of fused rings that has conjugated double bonds. This conjugated structure is involved in energy absorption. So pair. So there's two fused rings, conjugated double bonds. And if you compare it with pyrimidines, it is only composed of one ring. Okay. So how do we remember which ones are purines and which ones are pyrimidines? For purines, just remember pure silver, pure AG, pure adenine, guanine, purines, pure silver. And then the rest will be pyrimidines. So you have the cytosine, thymine, and uracil as pyrimidines. But earlier, when I was in undergrad, I have one professor who shared with us how she remembers the pyrimidines. She said that the pyramids ay nasa city, so that's cytosine and thymine. So if that will help you remember, so ang pyramids ay nasa city, pyrimidines, cytosine, and thymine. Okay. So in the structure earlier, you have seen that these nitrogen-containing bases are attached to the sugar. So where is the point of attachment? So for the attachment in the sugar moiety, the 1' carbon of the sugar moiety, whether it is ribose or deoxyribose, is attached to the nitrogen at position 9 of a purine. So where are those numbers? So these are the numberings. So this is the position 9 of the adenine of the purine. So this is where carbon 1, the 1' carbon of the ribose or deoxyribose sugar is attached in adenine and guanine, but for pyrimidines, it is attached to the nitrogen at position 1 of a pyrimidine. So this part right here. Okay, so this is how they are numbered, and this is how the nitrogen-containing bases are attached to the sugar moiety of the nucleotides. Let's take a look again at our nucleotide structure. So we have discussed the pentose sugar. So we're done with that. We have discussed the purine or the pyrimidine base. Now we will take a look at the phosphate group. So nucleosides ang tawag if there's no phosphate group, if it's just the sugar moiety and the nitrogen-containing base. But if there's a phosphate group, we call it a nucleotide. So nucleosides can have 1 or 2 or 3 phosphate groups. That's why we have nucleoside monophosphate if there's 1 phosphate group, diphosphate if there's 2 phosphate groups, and triphosphate if 3 phosphate groups are present. So in general, we call them NMP. And then if we're very specific with the base, you can call them AMP or GMP or CMP or TMP. So that's how we refer to the nucleotides. So these phosphate groups you will see here is the ones responsible for the acidic property of the nucleotide. And at cellular pH, these hydrogen ions are freed, leaving the phosphate group to be negatively charged. So the DNA is negatively charged, and you can attribute it to the phosphate backbone. For the naming of nucleotides, so we have the DNA and the RNA, right? So for the DNA, it's important to take note that it is deoxy. So again, if there's no phosphate group, it's the sugar and the nitrogen-containing base is present. And if the N base is adenine attached to the deoxyribose sugar, the nucleoside is called deoxyadenosine. If there's a phosphate group attached to it, then the nucleotide is called deoxyadenosine 5'. Take note of the 5' because the phosphate group is attached to the 5' carbon. Monophosphate if there's 1 phosphate group, diphosphate if there's 2 phosphate groups, triphosphate if there are 3 phosphate groups. Other name for the nucleotide is deoxyadenylate. Now, if guanine is present as the N base, the nucleoside will be called deoxyguanosine. The nucleotide is called deoxyguanosine 5' monophosphate. Monophosphate or di or triphosphate. Other name for the nucleotide is deoxyguanylate. Next, if thymine is present as the N base, the nucleoside is called deoxythymidine. The nucleotide is called deoxythymidine 5' monophosphate. And the other name is deoxythymidylate. Last, we have cytosine. If the base is cytosine, nucleoside is called deoxycytidine. The nucleotide is called deoxycytidine 5' monophosphate or deoxycytidylate. Now, if you look at the ribonucleotides or the RNA, it's almost the same except that there's no deoxy term because it's not deoxy, right? The 2' carbon is hydroxylated. So the nucleoside, if adenine is present as the N base, is called adenosine. The nucleotide is adenosine 5' monophosphate, right? It's just almost similar. Just remove the deoxy part of the name. And then the nucleotide is called adenylate. For guanine, as the N base, the nucleoside is called guanosine. Nucleotide name is guanosine 5' monophosphate. And then the nucleotide, other name is guanylate. If uracil is present, it's called, the nucleoside is called uridine, or the nucleotide is called uridine 5' monophosphate or uridylate. And then lastly, if the N base is cytosine, then the nucleoside is cytidine, and then the nucleotide is cytidine 5' monophosphate or cytidylate. So I hope madali lang yung i-remember, class, because it's almost the same for the DNA and the RNA. It's just that you put the term deoxy for the DNA and then the rest follows. Now, The nucleic acid backbone. So we have mentioned that there's a phosphate group at the 5' carbon, and also the 3' hydroxyl end can be phosphorylated. So the nucleic acid backbone you will see in this figure is composed of alternating sugar and phosphate groups. So this is the sugar moiety. Okay, so you have here the sugar, and then the phosphate group. So how are they linked? These nucleotides are linked by phosphodiester bonds in the 3' and the 5' carbons. Just like proteins, these are macromolecules that are directional. So it follows a certain direction. In our last lecture, we have discussed that in proteins, the direction that is being followed is from the amino terminal end to the carboxyl terminal end. For the nucleic acids, the direction that is being followed is from the 5' end to the 3' sorry, to the 3' end. But what is the 5' end, and which one is the 3' end? So again, 5', 3', that gives you an idea that we are referring to the carbons of the sugar moiety. Which one is the 5' end? The 5' end is, of course, the 5' carbon position of the sugar moiety. It would be a free or phosphorylated 5' end. So where is that? So this is the 5' end. So 5' carbon. It could be phosphorylated. And point of attachment. So let's move here. So this is one of the 5' ends. And what's the other one? The 3' end. The 3' end could be a hydroxyl group or a phosphorylated 3' end. So you have here the 5' end and the 3 to the 3' end as the direction. So 5', 3' attached to the 5', and these are the phosphodiester linkages. Okay. Now, you will notice that aside from being directional, these heteropolymers are also linear. So there's no branching in nucleic acid. These are also regarded to as heteropolymers. What does that mean? It means that the DNA or the RNA are composed of different repeating units. Different because what's the difference? The difference would be the identities of the nitrogenous species. Okay. Now this is the Watson and Crick model for the structure of the DNA. So in the Watson and Crick model, I know you are very familiar with this one. In their model, there's a double helix, 2 helical strands wound about a single axis. So these are 2 strands. So this one strand that runs in a 5' to 3' direction is paired with another strand that runs in the opposite direction. So this other strand is in a 3' to 5' direction. So that's why we describe the orientation of the model to be anti-parallel, because one is going in a 5' to 3' direction, 5' to 3', and the opposite strand runs in a 3' to 5' direction. Now you will see here the bases, which are projected inward but are still accessible through the major and minor groups. So I have here a model. Let me stop sharing so that you will see better. Okay, so there's no label for the phosphate groups and the ribose sugar, but we mentioned that there's an alternating sugar phosphate backbone. So let's just think of this backbone here as the phosphate sugar backbone. So we can think of this one as the ribose sugar, and then the phosphodiester linkages. And then in the middle, projected inwards, you will see that there are the bases. So you have base pairing, and we will discuss the base pairing in a little while. So this is a double helix. There's an axis. If you look at it from the top. So there's a single axis in the middle, and you will notice that it's like a twisted staircase or ladder. Okay, so we will go back into this later on. Going back to our PowerPoint. Next, we have the hydrogen bonding between complementary DNA strands. So in one-stone and quick base pairing, we know that A pairs with T and C pairs with G. So this is a stick diagram. And in this stick diagram, you will still see that there is the alternating sugar phosphate backbone. So you can still see that here in this portion. This doesn't show you the double helix, but it gives you a clearer picture of the base pairing. Okay, you can imagine here that the bases are placed or stacked on top of each other, just like in the model that we have here. So parang patong-patong sila, stacked on top of each other. And then please notice the ATCG base pairing here in boxes. I will just make the boxes red so that it will be easier for you to recognize. A pairs with B, C pairs with G. So why is it paired this way? A doesn't pair with any other nucleotide, with any other base, and G doesn't pair with any other base except for C. This is important so that there will be copying of the genetic material. We will see how important it is when we discuss DNA replication. But aside from that, another importance is to maximize the hydrogen bonds. So in this way, if A pairs with T, there's 2 hydrogen bonds, and C pairing with G will give us 3 hydrogen bonds. So that maximizes. With this kind of base pairing, there's a total of 5 hydrogen bondings with this purine pyrimidines. Okay? These hydrogen bonds between the nitrogenous bases are the ones responsible for holding the 2 strands together, and this contributes to the stability of the DNA along with other forces of interaction, between the stacked base pairs. Okay, now let's take a closer look at the strands. So here you will see the directionality again. So 5' to 3'. Let me just write it in this manner. So 5', you will look here, CAAT, 5'. We can also write it in this way, CAAT, CAAT, CGTCA. CGTCA. And then do not forget to write that on the last side is the 3' end. And based on this information, you know that we have discussed earlier that the opposite strand runs in an anti-parallel direction. So this one here is 3', and then this one other end is 5'. And since we know that there's ATCG base pairing, we can actually write what the other strand, what the basis of the other strand would have. So this is, since this one is C, then this will be G, this will be T, this will be T, this will be A, and then this will be G. G pairs with C, T pairs with A, C pairs with G, A pair with T. So you will see here that this is one of the features of the DNA that allows it to be replicated, so that if you use one strand as template, you can still produce the new strand. Okay? What else? So I guess that's it for the hydrogen bonding between complementary DNA strands. Next, we have different forms of the DNA that the DNA can assume. So these are structural forms. We have the B form, A form, and Z form. Now, for the Watson and Crick model, that's the bDNA helix, which is the most common form that exists under physiological conditions. So just take note of that. So this one, take note of this one, that this is the Watson and Crick model, the most common form that exists under physiological conditions. If you look at these different structural forms, there are a lot of characteristics, but I just want you to take note of three. So we have here the shape. Please remember this one. And then the handed bends, right-handed or left-handed, and the number of base pairs per turn of the helix. So these ones are the important things to remember. For the Z form, it's the narrowest, B is the intermediate, and A is the broadest. In terms of the screw sense, it could either be right-handed or left-handed. And then in terms of base pairs per turn of helix, B form will have 10.4, and Z form will have 12, and A will have 11. I will just stop sharing again so I can show you the model again and discuss the right-handed and left-handed helix. So how do we know if it's a right-handed or left-handed helix? Class, medyo nakakalito lang kasi I'm using the front camera, but this is my right hand and this is my left. So if you're looking at the helix, earlier we described it to be similar, or it looks like a twisted ladder, right? So if you imagine yourself climbing up the stairs or a twisted staircase, you can have your right hand on the rails. If that the rails is on your right hand side, then that's a right-handed helix. Okay, so again, this is my right hand. This is, you can imagine that this is the rail. I am coming from this part, going up, and the rail is on my right hand side. So this is a right-handed helix. Okay? And then if it's the opposite, if you can touch the rails with your left hand, in this case I cannot because it's on the right hand side, that's a left-handed helix. Okay? So that's it. Next, let's just discuss certain DNA sequences that are unusual in structures. Palindromes. What are palindromes? You may have encountered this before. It's not only used to describe DNA sequences. These palindromes are DNA sequences that read the same as backward and forward. So pag binasa mo daw siya pa forward, it's the same as binasa mo siya pa backward. So how do we explain that? So if you look here, this is T, right? T, A, G, C, A, C. If you look and read it backwards, it's the same. T, A, G, C, A, C. So that's what a palindrome means. Next, we have a mirror repeat. Mirror repeat is like a horizontal duplicate. So this is horizontal. If you cut it horizontally, it's a duplicate. So if you fold it, it's the mirror. It's like a mirror image. If you fold, so you have here T, and then you have here T here, and then again here T. So it's the same, right? A, and then A, and then G, C, A, and then CC. So that's what is being referred to as mirror repeats. In the DNA, since DNA can do base pairing, it's not only the opposite strand where it can do base pairing. It can also form a structure that it kind of loops with itself, because if it encounters a certain sequence that will allow it to form loops or this kind of hairpin structures, it can fold. It doesn't matter if it's not the opposite strand, it can fold on its own. Okay, so if this is one strand, you will see here that it's a 5' to 3' strand, but there are complementary sequences that you will see, like it has to be not only one class. Of course, it can fold, but during the folding, medyo dapat there are several bases, sequence of bases that can do the fold so that it will be able to form the structure. So ano bang ibig sabihin ng sinasabi kong folding? You will see here the sequence TGC, GAT. And we know A pairs with T, so this one can actually pair with that one. G can pair with that one. And then C can pair with the G in here, so this one. And then C can pair with the G here. A can pair with the T here. And A can pair with this one here. So it forms hairpin structure. So that's, for this kind of structure, it's important that the base again is not only 1 or 2, because that will not give you like a strong complementary base pairing. Since that's one strand, and if you imagine the DNA to be double-stranded, then definitely the other pair, the opposite strand, will have the complementary sequence. And since that sequence can also form hairpin, then that's what we call now as a cruciform structure. Okay, so this one is the strand that runs in a 5' to 3' direction that formed a hairpin. And then this one is the one opposite strand that runs in a 5' to 3' direction. But since there's complementary base pairing of this pair of strands, then the other strand can also have a hairpin structure and then form a cruciform. So it's like a cross. Next, we have secondary structure of RNA. So it's not only the DNA, it's also this kinds of secondary structure can be also possible with the RNA, because they also have, these molecules also have base pairs. So you have here single strands of RNA can also form this loops, internal loops, because this one doesn't pair with this one. So there's no base pairing here, but there are this portion right here, which can be utilized in the complementary base pairing of different strands of the RNA. So these are also possible with RNA. Next, we have mutations. So in the DNA sequences, there could be changes. In our discussion of proteins, last lecture video, we were talking about sickle cell anemia and sickle cell hemoglobin. And in that sickle cell hemoglobin, there's a change in the amino acid sequence in the primary level of organization of the protein. But since we know that the information that is stored in the DNA is what dictates the sequence of the protein, then this amino acid change has a corresponding base change. So these are mutations or changes in the DNA sequence which are caused by mutagens. These mutagens could either be physical or chemical agents that can cause mutations. There is a base pair substitution from the term itself. A base pair would be your complementary bases, one base that pairs with one another with the other, and this base pair will be substituted with a different base pairing. Okay, so this can result to a change in amino acid sequence, but this can also not result to a change in amino acid sequence. We will discuss more on that later. Next, we have insertions or deletions, or if you encounter the term index, these are additions for insertion, these are additions or losses for deletion of nucleotide pairs in a gene. So in the process of copying the genetic material, there could be mutations, insertions, or deletions, or if there's exposure to mutagens, there could be insertions or deletions also. But insertions and deletions are quite tricky to analyze because in this kind of analysis, you need to have a reference sequence so that you will be able to determine if another sequence has an insertion or a deletion. It could be relative to the reference sequence. So these mutations could have a disastrous effect on the resulting protein more often than substitutions do, because there could be changes in the shift, or there could be shifts in the frame of the reading frame, or ORF, open reading frame of the sequences. So it may alter the reading frame, like what I said, producing a frame shift mutation. This can also lead to truncation of the protein, so there will be no exact or complete amino acid sequence that will be available, or that will be folded into a 3D conformation that will allow the protein to do its function. We still have a lot of discussions on mutations. We have a lot of other examples that we will focus on towards the end of lecture number 9, that's on the central dogma of MolBio. So that's it for now. We will proceed with the discussion of metabolic pathways in our next video. So that's going to be about cellular respiration. Thank you for listening. I hope this has been a fruitful discussion. Thank you.